All right, good afternoon, everybody. Welcome back. I hope everybody enjoyed their lunch. They're getting up and walking around and socializing. So we have an action-packed afternoon today. So we're going to jump right in with our first session, Experimental Models of SATB2-Associated Syndrome, presented by Dr. Fish of University of Massachusetts Lowell, Dr. Fish is also a member of our Medical and Scientific Advisory Board. So Dr. Fish, thank you.
So good afternoon, everyone. It's always a pleasure to come to the SATB2 conference. I think this is my third one or so. It's hard to remember given the last 2 years where things have been a little bit crazy. So for those of you who maybe don't know, I've been working on SATB2 for, let's see, close to 15 years now or so. I've been studying it in different contexts. Initially as studying jaw development. And through the course of my research, I've become more interested in expanding our understanding of what SATB2 is doing during development in different tissues and how misregulation of SATB2 is involved in disease. So my contact information is there. And if you have questions, so you're welcome to ask questions during the talk if you want. It might be easier sometime, or after. And if something comes up later, please feel free to email me. So what I'm going to talk about today is kind of based on the research we do in my lab, a little bit closer. Is that better? Yeah, I have issues with projecting my voice sometimes. Mostly because I don't like the sound of my own voice. But anyway, so we'll talk about the research that we've done in my lab and some of the questions that we're thinking about addressing. And I'll touch a little bit on some of the other research that I'm aware of and things that we're facilitating. So we'll be really interested in feedback that families may have, because ultimately we are interested in serving the family needs. So I don't really need to introduce at all SATB2 and SATB2-Associated Syndrome, but I'm putting this here because this summary deals with, let's say, the major phenotypes of SAS, and later in the talk I'll get into some other, let's say, less focused on phenotypes that we're also interested in and trying to understand them and treat them. But of the major phenotypes are ones that are, broadly speaking, fall into two categories. So things that are related to neurons. So if we think about what cell types are affected in this disease, there are things that are associated with neurons and those that are bone or bone-related. And so in that context, we study SATB2 and its role in particular in these two cell types. And this has been, most of the work on SATB2 has been either in neurons. So this is a neuron, and that is a osteoblast or a bone cell. And so what are those cells doing, and how is SATB2 important in mediating what they do? So there's been a fair amount of research in that context. And what I would say, a major thing that we know that is consistent between those two cell types is, what is SATB2 as a protein? What does it do? So special AT-rich binding region 2. This protein binds to DNA and has two principal functions, and I'll get a little bit more in detail on this. But what we think that it does is it is contributing to chromatin modeling and creating these kind of loops. So sorry, that went forward. But so in the black is meant to be DNA or chromatin. And then we see that there are genes in blue and regulatory elements here in yellow. And so what SATB2 would do is organize these loops around the nuclear matrix. And when it does that, it puts these green or the blue parts, which are the gene regulation, in loops that are open. And so that allows those genes to be turned on and those proteins to be generated and do what they do. And then you can imagine that if this is looped in a different way, then those genes might not be able to be expressed properly. So these loops then would be specific to different cell types where the important genes for that cell type are open, if you will, or available to be expressed. So that is just kind of the background. There's already been a lot of research that sort of tells us that, right? That tells us SATB2 is important in neurons, SATB2 is important in bone cells, osteoblasts, and it's doing this by regulating chromatin and gene transcription. And so what I want to discuss today or cover a little bit are some of the major questions that we're thinking about and we're trying to answer now is, one, are all SATB2 mutations the same in terms of their function? And I think at least for a long period of time, we've been considering all SATB2 mutations to be loss of function. So no matter what the mutation is, it's causing SATB2 not to function the way it should, and therefore the cells don't have enough of SATB2 functioning normally. Now, is that the case, or do different mutations maybe perform somewhat good, or sometimes we have a mutation that is actually performing a negative function? Is that possible? So that's a major question. We're interested in what are the cell type specific functions of SATB2. So I've just told you two cell types, and SATB2, maybe it's binding DNA in both cells, but in terms of what else it might be doing and what are the specific genes or that other proteins that it's interacting with may be different. And that may be important because then the treatments that we would think about for a specific tissue might be different depending on the partners. So related to that is, are there specific interacting partners? And part of this too can help explain why even if two individuals have the exact same mutation in that whatever that mutation is doing, those two individuals might have slightly different what we call penetrance or occurrence of different phenotypes because the other interacting partners are different. So that's part of what we want to understand. I think a big thing that we're interested in are the specific cellular processes, because when we think about therapy, in many cases, especially in the case of a neuron, it will be really hard to add SATB2 to that neuron, to say, you don't have enough SATB2, we're just going to give you more. Based on what we know about SATB2 and how it works, it'd be really hard to do that. But if we know that the loss of SATB2 in that particular cell maybe disrupts a certain metabolic process, then we can treat that metabolic process. And so that's what we're really interested in thinking about. What are the next downstream consequences that might be more immediately treatable? And then I think a big key way that in particular we are, and I say we, my lab, but also other groups, and there's particularly this combined brain organization, which you might have heard about, which is a foundation. They're bringing together scientists and family groups that have neurological disorders and bringing them together to see if they can look at sort of shared symptoms or phenotypes that we can then address. And then it's no longer like one rare disease, but we have a big community that's addressing that. So what can we learn from other disorders that have similar disruptions, especially neurological issues that they've made more progress on, and that we can perhaps take advantage of that to help SAS patients? And then finally, what are some other phenotypes that aren't among those two major ones, like the neurological disorders and or bone disorders, that may also be important to address, and how can we maybe identify and find solutions for them? So to get at the first question and addressing this issue of are all mutations the same? So this is a image from a recent paper that was predominantly coming from Dr. Zarate, showing, just giving you an idea of an example of all the different types of mutations, and that they are really distributed throughout the protein. So this is a structure of the SATB2 protein, the different important domains, and you can see that these mutations are throughout the protein. So you might expect that a mutation that falls in a domain that is expected to have a known function might have a different impact than a different mutation. But so far, we haven't seen any obvious correlation between the specific location of the mutation and some phenotype. And that is also detailed here, just with an examples of a few different mutations in some of the different phenotypes, whether they have them or not. And so this variation may be due in part to the different mutation, but also, as I said, to some of the other interacting factors that may affect that individual. So how are we addressing this question? And I'm just going to talk very briefly about work from a different lab. And this is a project, Dr. Shinya Yamamoto, who's at Baylor College of Medicine. This work was funded by the SATB2 Foundation. And Dr. Yamamoto works in fruit flies, or Drosophila. And this model system has a lot of advantages. You might think, well, a fly is very different than a human, and in fact, yes, that is true. But many of the important developmental pathways are conserved, and in fact, much of what we know about disease mechanisms has been initially studied in Drosophila. They also work, they have a much faster, let's say, rate of development, and the ability to use certain genetic tools allow them to be very useful to understand certain questions. So basically, what his lab is doing is trying to assess the role, what these the different SATB2 mutations, do they all act the same? And so hopefully I won't mangle too much the work, but essentially what his lab is doing is starting with a few different mutations, and I think these ones identified here. And part of why these mutations were chosen is that they are ones that we also have iPSCs for. So the idea is that this research is synergistic and complementary, and that we use the FLY system to help us identify what variants might be interesting to pursue, where we might be overlapping, and how we can then set up our experiments in our iPSC or mouse models to further address some questions. So what I'm going to show you, I'll try and describe a little bit this work, but what the take-home message is that different mutations are in fact different, and that some of them have this loss of function. What we say, the mutation just causes SATB2 to not work, and therefore, in that case, that individual would have not enough SATB2. But there are some mutations that actually have, the mutation gives that protein a different function, and that different function is mostly negative, and that's what we call a gain of function mutation. So in this experiment, this is showing you Drosophila I, and basically the experiment was to overexpress or to make the normal developing eye have more of a certain protein and see what happens. And so if you just put SATB2 in and give it more SATB2, it causes the eye development to be disrupted. So you can see that there. So the comparison in this experiment is to SATB2. So if they use instead this particular mutation, the R389C, what they get is basically a normal eye. And so what that's saying is that that is a loss of function because it's not doing anything. So it's effectively saying you're overexpressing a protein that does nothing. On the other hand, this other mutation causes the fly to die. So that's obviously worse than what the SATB2 alone does. So this is an experiment that basically tells us that these two mutations are causing the protein to function differently. In one case, it does nothing. In one case, it's doing something else we don't understand. But this would then tell us, well, these are two example mutations that we might want to study further in a different system to understand how they're functioning differently, especially a gain-of-function mutation. Another thing that I will note about this is that, again, so these mutations in this case are different, but also this experiment done in a different cell type might have a different result. So there's one mutation that in overexpression in one tissue has a loss of function, and in another tissue a gain of function. So again, that tells us that what SATB2 is doing is cell type specific, and so we need to think about that when we think about therapies. Okay, so I think just moving on to trying to now understand some of these cell type specific mechanisms. I will briefly discuss some of the work that we've done looking at bone development, and we use mouse models, both actually in vivo in the mouse as well as in vitro in a cell culture system. And in that system, we used CRISPR to make mutations in the SATB2 gene. So we have a number of different mutations that we used. All of these are loss of function mutations. So an important thing that we've discovered in the bone cells is that SATB2 seems to be involved in proliferation, and in fact, that it's differently expressed in different cell cycle. So within a normal, healthy population, so if you look up, see, I already pressed the wrong button here. This upper left corner, this is a wild type, or a cell that has no mutation. And what is in green is just the DNA, so you can see the DNA in these different cell types. And then what is in pink is SATB2. So hopefully you can appreciate here that there are a bunch of different cells. This one is a dividing cell, and that the amount of pink, even in a normal, healthy population, is different between cells. So this is something that makes us wonder, why is that? It tells us that SATB2 is doing different things at different times even. And another important thing I'll point out here, so these 4 on the right are different mutations, and you can hopefully see that there's less pink, and in fact this one has no pink. And what we start to see are some misshapen cells, and in fact cells that these have just divided, just like these, but they kind of maintain these abnormal connections. And so we're trying to understand what is happening with that. So I'll actually just go quickly through this. This is all published, so if you're interested, you can find it there. That showing that the different mutations allow or inhibit proliferation of these osteoblasts. And so this is what we were thinking, is that part of the problem with generating bone is that they don't have enough cells. So the way bone is formed is the cells are depositing matrix or extracellular matrix. So that's what gives the bone its strength. And they just don't have enough cells. So one of the things that we were trying to do is think about how we could increase the proliferation of these cells. So what we just showed here is that we can artificially make a mutant. This is an in vitro system where we grow cells on the plate and we differentiate them. So we give them the nutrients that they need to go from a progenitor to making bone. And that takes about 21 days. And so normally, so this red, these red dots it's an indication of calcium deposition. So the more red, the more minerals you have. And what we were able to show is that if we just increase the number of cells, that even in the mutants, we can get bone that is equivalent to the wild type just by starting with more cells. So that was suggesting to us that it's not that these cells don't know how to make the minerals, they just don't have enough of them. So could we fix that problem? And this is something that we are currently working on. Another aspect of our research is to try to understand what is happening with some of these weird nuclear shapes. And what we currently think, so I told you at the beginning that SATB2 is involved in forming these chromatin loops. And that is important in the phase of the cell cycle where the cell cycle is G1, it's doing most of its work. It's producing the proteins, it's doing whatever its job is, if you will. But those cells, if they're proliferating, they need to divide and make more cells. And that requires DNA replication. And so it seems that SATB2 is also important in mediating this DNA replication. And so when there's not enough SATB2, the relationship or the integrity of the Mars may be disrupted, and that might be related to some of these aberrant nuclear shapes. So we're trying to understand more about how this is a cell type specific function. This is something that would only be happening in proliferating cells, how that might be regulated. Finally, another major question that we're interested in this work is related to what happens when you have less SATB2. So in this particular image, so again on the left are an example of wild type cells, so 2 different cells. And I'm showing you here these DNA loops, and this is a normal, what would be a normal healthy cell. And so SATB2 is the orange circle, and it's doing 2 things. So it's one, it's forming the loops on the nuclear matrix, and then it's also in bone cells, we know that SATB2 interacts with another protein called RUNX2, which is really important to make bone. It's a master regulator of osteogenesis. So you can see that SATB2 has these 2 important functions, and when SATB2 is at its normal level, all the cells sort of, SATB2 goes where it needs to go, and we have a coordinated cell population behavior. And this coordinated behavior is really important because in osteogenesis, you need to have a certain number of cells together to produce bone. And also in neurons, very important that the neurons are communicating to each other. So that they are coordinated is important. Now, a major question that we're interested in is, let's say in a healthy cell, you have 100 molecules of SATB2, but when you have a mutation and you have less, say now you only have 50 or 60, and there are 100 spots, right? Where do those 60 go? So you can see that in this example up here, cell number 1, so SATB2 is still there, and it goes to some of its normal places, but we have some mislooping, and maybe that mislooping occurs here. But in the next cell, the SATB2, just by chance, chooses a different 60 spots, and so you get a different mislooping. And so what this results in is unsynchronized or dysregulated population behavior. So that is one problem that we are trying to address. So what happens when you get dysregulated population behavior? And we're also interested in knowing, is there a preferential spot? So if you have reduced SATB2, is it just random where it decides to go and do its job? Or are there preferential spots that get filled first, and then there's a set of spots that just if they happen to be occupied, get occupied? So this is work that is currently funded by NIH to try and determine what happens when SATB2 is low. Okay, so I'm gonna skip because I'm looking at the time. That just shows that the gene expression is dysregulated, so we are fairly confident that that's what's happening. So getting more to what are the other cellular processes that we might be able to try and treat. So in the bone cells, we're looking at proliferation, but why is, when we say proliferation, the way we assess that is that we basically quantify cells at different time points, and we say there are less. So there could be less cells because they don't grow as fast, or because they die, some of the cells die, or they're differentiating early. There are a number of mechanisms that could cause that reduced proliferation. And one of the mechanisms is apoptosis, and we're investigating this mechanism. So what you're seeing here is the green bar is quantifying the number of live cells in a population, and the orange and magenta are quantifying cells that are either dead or dying. And this C8 is our mutant cell line, and you can see that there is an increased amount of apoptosis, although I will say that we know that apoptosis in itself is not the only mechanism that's causing these to have reduced proliferation. But what we think, so this is getting back to some of these weird nuclear shapes that we see with these cells that have SATB2 mutations. So we want to, we're investigating the role of oxidative stress in these cell types, and we use an assay that basically we can isolate the cells and stain them, and then the different stains, so in this case, the cell rocks is a measure of the activity of certain oxidative stress. And then the cytox, it's a dye that enters dead cells. So we can quantify if a cell is alive and unstressed, we'll just see it there in the bright field. But if it is dead, it will also be red. And if it's stressed, it will be green. And so we can quantify this. And what we see is sort of, this is what the data looks like, is these charts. So this is a flow cytometry analysis. And this is measuring the fluorescence, and it does it in several channels. So those cells that are healthy and unstressed are yellow, and they appear in this box. And then our stressed cells appear in these other two boxes. So our stressed, live stressed, and dead stressed are in these two boxes. And if you don't have the time to count each dot, the machine does it for you. And this might not look like much of a difference, but what we see is that the mutants have double the amount of stress cells. And it's still small, but this is under normal conditions. So what we did was we stress them out. And in that case, we used a drug to do that. And then we used, we tried different amounts, because even the healthiest cell, as you know as a human, even the healthiest person under a lot of stress will be stressed. But certain situations, we wanted to find a situation that would be a little bit of stress that wouldn't bother a healthy cell, but that might push the SATB2 cell over the edge. So in that case, it was that lowest amount there. And when we treat the cells, what we do is we treat the cells with this drug that causes them to be stressed, and then we do the same analysis. And what we found is that all of a sudden in the SATB2 mutant, we get a lot of stress cells. So what this tells us is that stress itself, or the oxidative stress pathway, isn't normally a problem, but if something happens, which it does in normal life, when a stress is there, then that's going to cause the SATB2 cells to respond in a way that will either kill them, in this case, more cells are dying, or just cause them to be stressed, which might disrupt some of their metabolic functions. So what we're trying to do is evaluate different drugs that now will reverse that. So we can treat with different drugs and see if, or especially ones that have been reported to reduce oxidative stress. So we have tried a few, and we have yet to identify one that works. However, we started looking at gene expression of genes that are known to be involved in oxidative stress. And so this was actually an honors project and a student that was working in my lab. And here showing you 4 different genes. And most of them, so in the yellow or orange is a wild type cell, and in the blue is our mutant cell. And then we have the ones that have the dashed line we have treated versus untreated with the stressed drug. And the important thing is that most of them are not different. So that says that in many aspects of stress or stress response, we don't see a difference between our wild type and our mutant cells. However, with this one specific gene, this NOX4, and we do see a difference. So we see both that SATB2, it's normally elevated, SATB2 in SATB2 mutant versus wild type, but then it becomes even more elevated when we stress the cells. And so this suggests to us, this is helping us identify specific components of the oxidative stress pathway that we might be able to target. And so this is something that we're trying to explore. So this is an example of bone remodeling. So normally bone is replenished. We have newly forming bone and then it's remodeled. So osteoclasts will remove some bone. And so we are trying to find the way. So oxidative stress causes, it induces osteoclast activity or more bone degradation, and it inhibits bone formation. So we want to try to manipulate this process so that we can actually try to encourage more bone formation and less degradation. So that is what I will say about the work that we're doing in bone development. Now, what about neurons? So at least in my lab so far, we haven't been doing a lot of work with neurogenesis, but now that we have the iPSCs, we're starting to pursue more work in this regard. And so what are some of the things that we're interested in? So, and I will get back to it. So this issue of oxidative stress or dysregulation of metabolic pathways, we're starting to think is a general problem with all SATB2 affected cells, all cell types. And so that is something that we are going to investigate in the neurons, how their metabolism and potentially oxidative stress is affecting them. But what I can tell you sort of what is we've seen in neurons, and this is an example from a mouse neuron, and this is also published. I'm still pressing the wrong buttons. So this is, so these were neurons that have been isolated and they're growing in a dish. But you can see here, this is a neuron, and you have these, basically these dendritic branches. And when a neuron that has been reduced for SATB2, you see that there are a lot more of these branches. And so that's just sort of shown here. And this is the type of thing that could potentially create problems with neurons communicating to each other, because they're going to be communicating through these extensions. So, and I don't have the data here, but this has also been seen in SATB2 or in human neurons on a dish in which SATB2 is reduced. So why is that happening? And is that something that we could potentially modify through a therapeutic treatment? This is an area of research that we would be interested in. I think the last thing I'll say on this is that getting back to this, what I mentioned at the beginning is that, I am just me. I have a small lab, and even though, I want to do as much as I can only do so much. So the way forward is to have more people working on SATB2, but also if we're interested in understanding neurodevelopmental disorders, can we tap into what we know from other neurodevelopmental disorders? And in fact, there's a lot of research on that. So one of the other projects that's in my lab is on a gene that's important in this 22q11 deletion syndrome, and I was just at a conference about that. And in that field, they have quite a bit of research on some of the neurological mechanisms underlying these different phenotypes, many of which are shared with SAS patients. And so the question is, can we utilize these other resources and some of the other, the knowledge that we've gained from experiments in those systems to help us form or design our experiments in a way that can help SAS patients? So that is something that we are doing. And in particular, there are a number of different mechanisms that we're interested in. So these different NDMA receptors is an area that we're going to target, and I don't see it from here, but we are definitely interested in oxidative stress and potential mitochondrial disorders. So those are areas of research that we will be pursuing in my lab. Okay, so the last thing that I want to talk about before I get a little bit to the iPSCs is what are some of the other phenotypes and that we might identify and sort of try to understand other mechanisms. So in particular, a couple of years ago when I was at this meeting, I was at a table with a bunch of parents and some of the other things that came up, and I remember the discussion of sleeping difficulties and things about hyperactivity, low body weight, things that fall in that potentially are related to a hormonal or metabolic dysregulation. It started to get me thinking about the thyroid a little bit, but just in general, do we know more about what is going on in the developing head or in the head where SATB2 is really important. So I'm going to actually skip a couple of these slides and go to this one that shows, so this is a mouse embryo. So this is a brain, these are limbs, tail, and all of this here in blue are this pharyngeal region, which is going to give rise to the jaw. So that will give rise to the jaw, as well as all of what is in your throat, your laryngeal cartilages, and as well as things like your thyroid, parathyroid, et cetera, tongue. So this is a section through that embryo, and you can see again in blue is the area where SATB2 is expressed. And I'm pointing this out because, as most of you know, that SATB2 has a major impact on the jaw and tongue, but is also expressed in the more caudal pharyngeal arches, as well as this tissue on the inside, this endoderm, that will give rise to the thyroid, among other tissues. And so here on this bottom panel, this is a SATB2 mutant mouse, and on the right, they have removed the cartilages from the throat. And you can see that here, this thyroid cartilage is essentially, it's missing in the SATB2 mutants, and these other cartilages are reduced in size. And in fact, one of the things that we're interested in looking at is the airway itself, and if there are issues with development of the airway, which might contribute to things like sleeping disorders. So what we're doing right now in the lab is a full phenotypic analysis of the entire craniofacial region. And we're using a number of different technologies. So we have the mouse embryo, and what you're seeing here, so this is a skeletal preparation. So it's a assay that looks specifically at skeletal elements, and all the soft tissue is removed. But we also can look at micro-CTs. So that's what is shown here. So this is a section basically through the neck region of mouse embryo that has undergone micro-CT, and this allows us to look at a lot of the soft tissues. And so what you're seeing on the right is the thyroid gland. And so I had a student who used these micro-CT images and was able to go in and quantify the thyroid gland, and interestingly found that the SATB2 mutants have larger thyroids. So that is just shown here, that you can see that there's a difference in size, a little bit left to right, but in general that the SATB2 mice may have hypothyroid or hyperthyroid issues. And this would be consistent with things like hyperactivity, high metabolism, et cetera. There is some evidence from, so these data were taken from lymphoblasts. I might be slightly off there, but this is a work from a collaborator that measured various different metabolic outcomes in cells and showing that, that the SATB2 mutant cells, they have a unique metabolic profile, and in fact, response to different hormones is also dysregulated relative to the healthy individual. So we're interested, and this again is part of evidence that is pointing to a general dysregulation of metabolic activity in cell types that may be something that we can potentially treat or target as a treatment. And sort of the last thing that we're looking at right now, as I said, part of this whole phenotyping of the mutant mouse is we're looking at the trigeminal nerve and the, particularly the temporalis, but other muscles that are involved in chewing. So what we've noticed is that, so in the mice, when we say a mutant mouse, usually it has 2 loss of function alleles, so no SATB2, which is different than human patients, which have 1 normal allele and 1 mutant allele. And in general, we call it heterozygote mice are fine. We keep them, that's how we use in the colony, we breed them. But what we notice is that I would say it's about 1/3 to 1/2 of those mice die before weaning. And they die because of failure to thrive. And so what we don't know is this a metabolic issue that they're getting all the food that they need and they just don't have enough to develop, or is there something more related to their feeding? So their ability to regulate their muscles for suckling. So the mice, first 3 weeks of their life, they're suckling. And we think that it's possible that there's some problems they have coordinating the muscles in their jaw to get enough food. So this is something that we're also investigating. So just to show that these muscles and how they're related to chewing, and the trigeminal nerve, so the trigeminal ganglia here, and this nerve which is innervating much of the face and jaw, and so really important for sensation, facial control, and chewing. So we do have evidence, and I didn't show it because it's very, very new, and I want to be certain of it, but that suggests that there are some issues with the trigeminal nerve in the SATB2 mutation. So I have 6 minutes, a little more than 6 minutes left, and so I'll just briefly tell you about sort of where we're at with the iPSCs. So the foundation, through generous support of families, was able to help us work with Cedars-Sinai to generate these pluripotent stem cells. So we had tissue donations from 3 patients and parents that we used to generate these iPSCs. And so we start with so there was skin tissue, so that's the fibroblasts, which are shown here. So this is what they look like on a dish. So basically, we received this piece of skin tissue, and you put that piece of skin tissue on a dish with media or liquid that has nutrients, and those cells will migrate out and they will grow. And they will grow for a very long time. So we have these cells in the lab, frozen. So my major role with the SATB2 right now is housing and maintaining these cell lines. So we have the fibroblasts from the patients as well as in parental controls. And so what the core facility did was they have a reprogramming. They reprogram these fibroblasts to induce pluripotent stem cells. And those cells look more like this when they're growing on a dish. And so we have 2 separate lines. I think that's true, 2 separate lines for each of the patients and maternal controls. And so the ones that we have are here. Now I will say that there are other labs that have, so at least 1 other patient-derived line that I'm aware of, and then there are a number of labs who just used CRISPR to knock out SATB2. So some of the data I talked about where we saw neurons were from CRISPR-generated neurons. So these lines are housed in my lab, and they are available to researchers anywhere. They just have to contact Allison. So, and then there's a form that is filled out, and one of the major things that we're interested in is we want to make sure that those labs are, number one, performing relevant research, and number two, there's no overlap, because we want to make sure that we're maximizing our resources and that we're not competing, but we're rather building knowledge. And there are at least 4 labs now that have these cells and are performing research on them. So just the last thing that I will say about this is we've started work on them, and most of what we've done so far in my lab is just to get the cells where we can have them up and running. But we have done, a few analyses. I don't know how clear it is. We, again, in this case, our DNA is blue, and then we have the pink again is our SATB2. And so on the left here, this is a patient, and it's a little bit, I don't know how clear it is, but hopefully you can see that there's a lot more pink or red, and this is the maternal line. So these are from the iPSCs, but still you see that even within the maternal line, there's variation. And that gets back to this point I made earlier that SATB2 seems to be really important in these highly proliferative cells. And in fact, in the iPSCs, we've looked at SATB2, and here you can see, so in this case, there's a lot of red, that the iPSCs, which are growing at the edge, those are the cells that are really high in SATB2 levels, and those that are more central and less proliferative have lower SATB2, and that's even in this maternal. And of course, the patient has a lot less. And why is this important? So we initially started because we were focusing on bone research, that we were trying to differentiate these cells into mesenchymal stem cells and see if we could repeat or follow up some of our work that we did in mice in the human cells. But the differentiation process in mesenchymal stem cells is a lot of proliferation, and these cells, the patient cells, just, they wouldn't grow. And so we spent quite a while trying to make them happier so that they would grow, but we've, for the moment, decided we're gonna just switch anyway. There seems to be more interest in pursuing the neuronal work, and so that's where we're going forward. But this is an interesting aspect of, just to say things are going to come up with these cells that we will need to address. And then I think the final slide I have is just thinking about what are the therapeutic strategies. And as I said from the beginning, one way is just, especially if the mutation is a loss of function mutation, try to find ways to increase SATB2. And mostly that would be by trying to understand what turns SATB2 on, and there is work known on that. And that will work in a loss of function. If a mutation is a gain of function, we wouldn't want to do that because that wouldn't help that individual. On the other hand, we have these more systems approach where we can try to understand the actual processes that are dysregulated and try to treat them. And we can really, in that case, look at shared disease mechanisms as a way to do that. So this is just my lab and some recent iteration of it. It's changed a little bit since then. And some of our collaborators, and thanks, of course, to the SATB2 Foundation and our Lindsay Panther, who was at Cedars-Sinai and who is, really helped us a lot with getting these cells going. And of course, Yuri and others. So, and thank you. And again, if I'm open for questions, but you can always email me as well.
Thank you so much, Dr. Fish. This was really informative. So I was wondering, like, in terms of the research that you had done, right? So have you looked at like the brain itself, right? Is there hypometabolism because of SATB2? And the reason I ask is some of the stem cell therapies that are around there, they work towards, I mean, they induce the same stem cells, and the intent is, right, they would work on the hypometabolism activity there. And again, it's not proven, so just wondering, like, you know.
So your question is, have we looked at the neurons for it? So at least, no, we haven't. I know there are labs that are doing several outcomes, like electrophysiology or other aspects, but in terms of have we looked at neurons yet for their metabolic output? Not that I'm aware. That's one of the things we want to do. And in fact, so I was looking at that earlier today. How can we do that? What are the best ways to measure these specific outcome measures? Yeah.
Yeah, I mean, we've done, I mean, we can talk to you afterwards, but PET scan is one of the things that people have talked about.
Yes, yes. So for sure that, and as well as looking at glucose uptake and other measures. Basically, in that way, glucose metabolism is a way that we'd want, and through the scans as well, yes. So that's just a question of, you know, working with someone that has access to that technology and getting them the materials. But yes, we 100% want to do that. Yeah. Okay.
We have a few questions online. The first is from Katie. My son had his parathyroid hormone tested at age 5. Nothing was flagged abnormal. How often should we test, or what other testing should be ordered to check thyroid function?
Well, so they can do blood. So that should be something that they could test in blood, and they could look at thyroxine levels. So there are levels that they could test in the blood. And if there was a case, so that should be pretty easy to test. How often, I don't know. I'm not, I don't know that. But the doctor should know how often they could test that. And then if that is the case, that should be a fairly easy treatment for certain symptoms. Yeah.
Okay, awesome. And then lastly, and for those online questions that we don't get to, we'll make sure we follow up after. So the last one is, how would one know if our kiddo has a mutation resulting in a loss of function or a gain of function, genetically speaking?
Right. So we have to test them all. And so it's another thing that I didn't mention. So I showed the figure where we have SATB2 that is binding to DNA specifically, or if it's binding to another protein. And you can imagine that the way it binds to DNA, it's using a different part than when it binds to a specific protein. And so if you have a mutation that affects one of those binding areas, then it would have an impact on one function versus another. So, and then that might be different from cell to cell. So basically, the plan, at least, is, you know, what we would like to do is get Shinya to make all of the Drosophila models and then test them there, and then we would do cell type specific testing on follow-up on the interesting ones, or the ones that have different, you know, loss of function, that's going to be already highly studied, but anything gain of function we would study in a different cell type. But yeah, I mean, at this moment there's no way, we don't understand why one mutation is loss of function and not, at least related to where they are in the domains. Yeah.
Just a quick question.
Yeah.
You said that oxidative stress makes it worse, you know, like you were stressing the cells. What if the oxidative stress decreases, I don't know, like with the diet, antioxidant diet, or maybe improves the behavior?
Exactly.
Or...
So, and in general, that's something that we could say for everyone, you know. Oxidative stress is known to, just in general, lead to disease and aging, right? So these are things that cause aging and disease. So we should all, you know, try to reduce our oxidative stress. But yes, it may be that there are more direct methods that we could use in certain, especially for certain cell types that we could target if it came to that. But yes, that's the idea, reduce the oxidative stress, and hopefully that helps in general. All right, thank you.